just go over every time. It's not just for your test, it's also for your own personal growth and learning. So please stop me if you want me to clarify some points, but all this presentation should be up on Blackboard or the portal that you use for assessing, like for the exams, for reading for the exams. So what is the nervous system? So what is your idea about nervous system? What do you think is nervous system all about? Any answers? So I think the PowerPoint slide here is summarizing it, meaning the nervous system is an excitable system. So nervous system is comprised of nervous tissue. So what are the different organizations? So first we have cell, then we have tissues, then we have organs, and then we have organ systems, right? So what do you think is nervous system? When we say system, what is it that we really mean? Which category does it belong to? Organ system, right? So it is made up of tissues, nervous tissue, but it is also made up of other tissues, which we will talk about a little bit as well. So the function of the nervous system is communication. What's the meaning of the word excitable? Excitable means that if there is a stimulus, for example, if I pinch you, you're going to cry, right? So that is excitable. That's the function of the nervous system. And it is found in all multicellular organs except sponges. So it is found pretty much everywhere. So if I studied the nervous system of a giant squid, which has big axons, I would be able to tell how it is working in a human being. So it's comparative always. So because it's not always possible to work in human beings or human carcasses, right? So we study other organisms to give us a clearer picture of what it would be in us. So what is the important word here? That there is an electric current which goes through our body, and that helps us communicate between different parts of the body as well as with the environment. Make sense? Let me know if there is something that is new to you and I want to go over again. So the giant squid axon looks like this. So as you can see, it's giant. So its one axon is pretty big. Now what is the functionality of how can we take advantage of that? It's very easy to study a giant squid axon, and then we can, most of the advancement in science in this area has come by studying the giant squid axons. So again, the function of the nervous system is stimulus is external and internal, and it processes information, and you have a cellular response to that external stimulus once that information gets processed. Make sense? So what are the type of neurons? So there are mainly, now how you classify the neuron, that would decide what are the different types. So one of the ways that you can classify it is by its function. So whether it has a sensory function or it has a motor function. So in this case, this is a typical sensory neuron which is collecting information from an animal's environment using these structures called the dendrites. It's almost like hands, pick up the information, and then it is processing it in the cell body, the nucleus, and then it is sending that information to the axon and passing it along to the other neurons or to muscles, and we'll see that later. Next is the types of, in the type of neurons is motor neuron, pretty much a similar structure, but like I said, muscles, usually the sensory information is like, for example, tactile information or auditory information, that is sensory. And that is sending the information to the central nervous system, that is the CNS, which is the brain and the spinal cord. So that is the function of the sensory neuron. Whereas the motor neuron takes the information from the brain, sends it to different parts of our body. That can include muscles. So what I said before was sensory information can send the information to muscles is not right, because it can only be done by a motor neuron. So during this presentation, if you catch me saying some things which is a little different from what you have known before, let me know, because that way we are learning, right? So I have a question on the formative assessment, which is asking what are the two different types of neurons. So based on what you have heard now, can you please write them down? And you are allowed to discuss with your neighbors. If you want to move and come closer to somebody, if you are sitting by yourself, you can come closer and you can discuss and write down the answer. Yes?
Is this being graded? Because I don't think it was passed around entirely.
Uh-huh.
I don't have one of those formatted.
Oh. Does everybody have the formative assessment paper? Where did it stop last? Oh, okay. Who all didn't get it? So just pass it around. I think there's many. So go. And I have more over here, so if you didn't get it, please come get it from here. Now, I'm not grading you on this. This is for your own learning, like I said at the beginning, but it would help you because you can keep it for your future reference. All of the answers to the questions would be up on, on the learning portal, so yeah, on Blackboard, so you would have this information with you. But right now, what I want you to do is to think about it because I kind of talked about it a little bit. So what do you think are the two different types of neurons? It was on the previous slides, right? So write the answers down, and then we can see if we got it right. So the two different types of neuron is sensory and motor, and the different parts of a neuron would be the dendrite, the soma, and the axon. Now, here I have just got them listed. But can anybody volunteer to say what are the functions of the different parts of the neuron? So I know what you got because right now I won't be able to check the answers, right? So what do you think is the function of dendrite? Right, receive the stimulus. How about the soma? Yes, so it processes the information. And then how about it's not just processing the information. The soma is also kind of like generating that electric current so that it can pass along via the axon to the next neuron. So we have the next one is the axon. So I kind of gave you the answer. So what do you think is the function of the axon? Good job. So it's like she just said, it's a highway for the electrical impulse to travel down and move to the next other parts of the body. So next is what are the different types of nervous system? So we need to classify things, right? Otherwise, it's also random. So when we are classifying it, what we see is the central nervous system composed of the brain and the spinal cord. So think of brain like the king. Why? Because you need to protect the king, and the king is a very important function. It has a lot of administrative functions. That's somewhat like the brain. That's what, how the brain works. So we have also the spinal cord. So let's say if there was fire in here and I touched it, that information is going via my sensory neurons and going to the spinal cord and then goes to the brain. And then I immediately remove my hand because of that stimulus, right? So those are the major. So central nervous system's job is to, like somebody just said, process the information. And then what happens is there is peripheral nervous system whose job is to take that information from the environment. So what I mean by that, so if I have got nerve endings in my skin, that would be part of the peripheral nervous system. So pretty much everything but the brain and the spinal cord would be the peripheral nervous system. Then you could also divide the motor pathways into different ways. One is the somatic nervous system, which is the voluntary motor pathways. So when I remove my arm from the fire, what pathways do you think are involved? Is it voluntary or is it involuntary? Involuntary or voluntary? How many involuntary? Okay, how many voluntary? Okay, why do you think it's voluntary? You don't have to do it? Yes. So the answer is both. It is both involuntary and voluntary. Now, once you get the sensation, your reflex action, which is involuntary, is to move your hand away. But what if you are a circus person and who wants to show some tricks and you kind of know that I can bear this much amount of heat, and so that's a voluntary action to not move your hand away from the fire. Make sense? So it is both voluntary and involuntary. Now there's also within the autonomic nervous system, which is kind of the involuntary stuff, there's something called the sympathetic nervous system, the other is the parasympathetic nervous system. So sympathetic is fight and flight. So if your neighbor's dog is coming, chasing you, you want to fight or fly away from there. So that would be the sympathetic nervous system. How about the parasympathetic nervous system? So you have had a good meal and now you want to rest and relax, and that would be the parasympathetic nervous system. Now what is reflexes? Now I just talked about a reflex to you, that you're moving hand away, that's a spinal reflex. Now what is reflex? They can generate a response to the stimulus without need for processing of the signal by the brain. So when I moved the hand away, do you think it was my brain that was, had something to do with it? Most likely yes. But if I give you another example, let's say if I have a hammer and I hit your knee, you would give a knee-jerk reflex, right? So in that situation, what is happening is you don't need the brain to make that, that reflex. It's only going via the spinal cord, and then you have an automatic response to that. So that would be pretty much what reflexes look like, where no brain is involved. So this is something that is just going via the spinal cord, and you have a response right away. So the motor neuron is coming from the spinal cord. So there is a sensory neuron which is going to the spinal cord, and then there is a motor neuron coming out of the spinal cord to those muscles or whatever that may be, and you have a movement, and that would be the type of movements that falls under this category, reflexes. It's a no-brainer. So why do you think it is important? Because it's fast. If it had to go all the way to the brain and come back, it would take more time, right? So if it is real quick and you need an immediate response to danger, that would be, a reflex, because it doesn't need to involve brain. So I have another question here. Again, if you don't have a formative assessment paper, you can come pick it up from here anytime. So your neighbor's dog is chasing after you. Which part of the nervous system would get turned on? So I kind of gave you the answer, but you can discuss amongst yourselves, but write down an answer that you think is most appropriate for it. Okay, so the answer is the sympathetic nervous system, and hopefully you start running away from the dog. So next is dendrites receive external stimuli. Now this is a little bit complicated concept, and I want you to work with me on this because it's not that easy to get this information. So as you can see, it is having 3 steps. So when it's saying the dendrites are receiving the external stimulus, the first thing is the receptor proteins within the cell membrane of the dendrite respond to the stimulus by briefly opening up little channels. So we are talking about electric current here, right? So electric current can pass through your body. So electric current involves what? Some charges, correct? So that is your basic knowledge from physics, or your background knowledge can tell you that electric current means there is some amount of ion charges that are involved with it. So these channels that we are talking about are ion channels. So whenever there is a stimulus, what happens is that there is a charge difference across the membrane which opens up these ion channels, and what happens is some sodium ions, which is positively charged, gets inside. So it opens channels and allows the charged ions, which is most likely the sodium ions, which is positively charged, that enters and momentarily alters the electrical charge within the cell. So usually inside the cell we have more negative charges, and outside the cell we have more positive charges. But when that stimulus comes in and it opens the channel, it lets in those positive charges and makes the inside positive. So signals from all of the dendrites of neuron coverage is, so most of the neuron will be covered, And then if the sum total of this signal coming is positive significantly, then the cell initiates an action potential that travels down the axon. How many of you have heard this word action potential before from high school or other place? Cool. Would you want to explain? Somebody want to volunteer? What is action potential? You don't have to. Go want to? Okay, you don't want? Okay, that's fine. So what is action potential? Action potential is a stimulus. So it's a movement of ions that changes the charges. I wonder we have, oops. Okay, here. So I said the inside of the cell is usually negative, right? So it can be anywhere like minus 80 millivolt or minus 70 millivolt depending on what type of cell it is or what species. Can everybody see it, or is it too light? Too light? Oh, never mind. So I'm going to just go with, oh, thank you. Thank you. So in here, we are seeing the resting membrane potential. Can everybody see? Okay. So here is the resting membrane potential of minus 80 millivolts. And then what happens is come a stimulus. The inside is now going to become more positive. So which way does it go? It goes like this. Goes up, right? Up is more positive. And then if there is enough of that stimulus received and it reaches the threshold, then it goes up. And then this phase is going to be called a depolarization. And then once it can go just as much as it can go, that means all the sodium channels are open, and then the charge would be somewhere like about 50 millivolt. Again, this is not a fixed number. It can be varying depending on what species we are talking about. So once it reaches that, then it falls down, and that process is called repolarization. And the repolarization is also happening because of opening of ion channels, specifically potassium ion channels. And when that potassium ion channels open, because there is a lot of potassium inside and not outside, what happens is potassium ions like to go outside. And if potassium ions are going outside, what do you think would happen to the inside? It's going to become more negative because potassium is positively charged. So there is a potassium which is positively charged. There is a sodium which is positively charged. During depolarization, sodium, which is more outside, less inside, is going to come in. Okay? So making it more positive inside. So that's depolarization. During repolarization, potassium channels open and they go outside, making the inside more negative. That's a summary. And now this is what an action potential curve would look like. And this is generated because of differences in ions, which is pretty much what our electric current is, right? So that's how we have the stimulus being transmitted either from outside our body or inside our body to different parts, to different other cells, okay? Now the axon propagates a signal. So in this, where do you think the signal gets generated? So the signal is generated somewhere over here. So we have a lot of ion channels at this place. So if there is enough amount of electricity being generated, enough amount to cause an action potential, then this neuron is going to fire. And that action potential is going to travel down this axon. Now you see that this axon has got some wrappings here. These white wrappings are basically nothing but fat, and they are called myelin sheath. So why do you think we need an insulator like fat, like myelin sheath in here, all around the axon? Yes. Right, pretty much. So we may have heard, we may not have heard, we know about conductors and insulators. So think of axon like a conductor. And think of the fat, the myelin sheath, as an insulator. So what it really does is it doesn't allow that information to get distorted, and a pure signal is being transmitted because of the help of this myelin sheath. The other thing is, at these nodes of Ranvier, which are like the gaps between the myelin sheath, we have a lot of channels, voltage gated sodium channels, which helps in making those, opening up, and therefore the action potentials are kind of boosted over here. So the information kind of jumps. So the stimulus jumps from node to node, and that is called a saltatory conduction, and that helps in faster propagation of the signal. So that's just a way we have, not just human beings, but other animals have evolved to, transmit the information really fast. So the myelin sheath prevents the action potential from weakening as it travels down the axon. And in here, like you see, this is the nodes of Ranvier, and we have positively charged ions coming in, so it kind of boosts the action potential, and we see more of these depolarization, where the inside becomes more positive. So neurons in the brain. Now, in your brain, you might have seen, oh, this person has a lot of gray matter. It's very intelligent, right? But there are these memes which go across. So what is a gray matter? What is a white matter? So the gray matter has the nucleus, or the soma, which we talked about before. And the white matter has the myelin sheath, because that you saw the myelin sheath was white, right? So it has those axon terminals. So the gray matter has got the nucleus, and the white matter has got the axon terminals, which has the myelin sheath in it. So that makes it gray, or that makes it white. Now inside the brain, what happens is, you can see this picture over here. Which one do you think is the white matter, and which one do you think is the gray matter? So if this is like, if you slice the brain and stain it, this is how it would look like. So if I'm considering this to be the inside of the brain, and I'm considering this to be the outside of the brain, so where do you think most likely the gray matter would be? On the inside or on the outside? On the inside, right. So on the outside, we have got the white matter most likely, and then we have on the inside, we have a lot of gray matter. Okay. Now that may not be the case if it was a spinal cord. Okay. Now in here, what is happening is the myelin sheath, the fatty coating that surrounds axons are white. So areas of the brain rich in axons appear white. Brain regions rich in cell bodies and dendrites appear gray. So just know that there is a white matter and there is a gray matter, both in the brain and in the spinal cord, and they're in reverse direction. If it was in the brain, one was inside, the other one was outside. Okay. So in the case of spinal cord, we see that it has a butterfly-like structure, and then it has got columns of white matter which goes all the way, to the brain. So that's, a little bit more complicated. I'm not going to go too much into that, but I'm going to have a question here which should be, answerable now that we have talked. But if you want, you can discuss it amongst yourself or ask me a question if need be. So where do you see the impulse or the action potential is generated, and where do you think the action potential travel? So in this question, I'm asking which part of the neuron, in which part of the neuron, so whether it is a soma or whether it is the dendrite or whether it is the axon, which part of the neuron would you see this impulse or action potential generated? Where do you see that generated? And where do you think the action potential travels? Do you want me to go back to a previous slide to show you? Okay. Okay. So these are dendrites. This is the soma or the nucleus. This is where the axon starts. So it's also called the axon hillock. And then we have the axon over here. Okay. Got it? So this is the axon hillock. So going back, the axon hillock is right over here. So it's a part of the soma, but it's where the action potential is being generated, and then it travels down the axon. Then comes synapse. So once that now we are talking about electrical currents, electrical signals being transmitted. Now how about this picture? In here we have what we see here is something called a synaptic cleft. Now when we talk about synaptic cleft, we are really interested in the chemical transmission aspect of it, and we are going to go more into the details. So when the action potential reaches the axon terminal, neurotransmitter filled vesicles fuse with the presynaptic membrane, releasing neurotransmitter molecules into the synaptic cleft. So what is a neurotransmitter? What do you think is a neurotransmitter? Do you have any idea about it? So I told you it's a chemical synapse, so neurotransmitter must be a chemical. Now what do you think that chemical is doing? So what the neurotransmitter is doing is taking that electrical energy, converting into chemical energy, transmitting it on to another neuron or to a muscle, and thereby transmitting the signal. Now how does that work? In this picture you can see some vesicles. Now these vesicles have got those neurotransmitters, and then what happens is when the action potential comes by, these neurotransmitter containing vesicles would fuse with the membrane, and this is called the presynaptic membrane. And this is the synapse, and it would allow the neurotransmitter to travel via the synapse to the postsynaptic membrane. In this postsynaptic membrane, you would see some receptor proteins like over here. These receptor proteins most likely are ion gated, so they're voltage gated and they're ionotropic receptors. They could be metabotropic, and I'm not going to talk about that. I'm just going to talk about the ionotropic receptors, meaning that these receptors have ion channels, and these ion channels are going to open, and now what you see is these the sodium ions are going to come inside. So what really happened in here is you have these neurotransmitters being released, and these neurotransmitters come to the synapse and they go to this receptor proteins. When this receptor binds to that neurotransmitter, which is a ligand, what happens is, imagine this to be a gate. That gate opens wide, opens, and then what happens is the ions are going to come inside. And when the ions are going to come inside, that would depolarize the membrane. When obviously there's enough amount of ions coming in, that would depolarize the membrane, and it would now in turn you would have the action potential. Make sense? So in here what we had is there's a chemical transmission. So the neurotransmitter is the chemical which had that information, that stimulus, that electrical stimulus, and that is being transmitted down. Too abstract? Do you want me to repeat? Tell me if you didn't get it. I'll be more than happy to re-explain. So you got it, right? So can I ask you a question? So what is this neurotransmitter? Is it the receptor or is it the ligand? These neurotransmitters that are coming through via the synapse, is it a receptor or is it the ligand? What is a ligand, guys? So imagine this to be a receptor. Here comes something, like some chemical, which I'm calling the ligand, and then the ligand binds with the receptor. And what happens is, obviously it was like this before, now the shape of that receptor ligand binding changes the conformation, and in this case would open the ion channels, right? So my receptors are on the postsynaptic membrane, which is right over here, the postsynaptic membrane. Now incoming are what? The neurotransmitters, right? So the neurotransmitter is my ligand, right? So we have the ligand that is binding to the receptors over here, changes its shape, lets in the ions, which in this case is the sodium ion, and that would make the inside of the cell more positive. Make sense? Now, how are you going to apply this to your life? Why is this even important to you? Why do you need to know in a non-majors course? Why is this going on? Because if you go out in the sun, the sodium in your body can go down, like generic sodium, you're sweating, sodium leaves the body, and that's not good for you. Why do you feel so tired? Is because you are now becoming more unable to have these action potentials, because in order to have those action potentials, you need those enough sodium in your body. And in case these channels are not working the way it is, if there is a problem either in the receptors or in the ligand, which is the neurotransmitter, you have a problem. And we are going to go more into this scenario later on. Okay? So I just given you one situation to your own life why knowing this information might be useful, right? So I have another thing over here. It's a picture of Rachel Zoe. Not too familiar with her. But what is the difference between the two pictures? Obviously, one. Yeah. So what did she do to not have wrinkles? Botox. What is Botox? It's a cliche. We hear it everywhere. Cosmetic surgeries and Botox, right? So what is Botox? How do you think that works? So all the information that you need to, it has already been given to you, and you could answer this question with that information. Why? Because Botox, and you see what is written here, by preventing the acetylcholine release, botulinum toxin Botox paralyzes muscles in tiny amounts. This can reduce wrinkles. Now, I think when I say I give you all the information, I may be overstating. So what is the acetylcholine in here? It is a type of neurotransmitter. Okay? So acetylcholine is a, I'm writing NT here, it's a neurotransmitter, and ACH is acetylcholine for me. Just want to write shorthand here. So by preventing acetylcholine release, so let's get back to the picture. So this is what? This is the axon terminal, and that had those neurotransmitters in here. Comes an action potential. What happens is this neurotransmitter gets released, and they are now out into the synapse. This was the synapse, and then there was a receptor, and that neurotransmitter would bind to that receptor and would open the ion channel and would let in sodium, right? When that sodium would come in, what would it do? If it was a muscle, so imagine this is a presynaptic neuron, this is a postsynaptic neuron. If this sodium comes in and it does contraction of the muscle, so we can have muscles in here. So we have muscle cells in here. So whenever that neurons have that information, they could either give that information to another neuron or they can give it to a muscle. So in this case, let's say there is a muscle in here. So usual circumstance, if there is an action potential coming in and then there is that information being passed through this synapse and it goes all the way to that muscle cell, the muscle would contract, right? So what would happen over time? Your muscles would keep contracting. Imagine too much contraction. If you over exerting your muscles, what would happen? It's going to get loose, right? You're going to start having wrinkles. But now what is going on is this acetylcholine. So there can be different type of neurotransmitters. So let's say one of the neurotransmitter is acetylcholine. So this neurotransmitter here is acetylcholine. Okay? Now that came in and it allowed the sodium ions to go past it, and you would now in turn have an action potential because of which you have a contraction. But in this case, this toxin, the botulinum toxin or Botox, what happens is it doesn't even allow these acetylcholine release. Can everybody see this picture? I'm not so sure because of this. Can everybody see the picture? Okay, good. So if this acetylcholine is not being released, what do you think would happen downstream? Sorry? Wouldn't contract. So in turn, you're not going to. Yeah, it wouldn't wrinkle over time. But what can be another side effect of that?
Limited facial expressions.
Yes. What else? So imagine your body is not in isolation. Like if every part is connected. Now, if you are trying to only act on your facial muscles, like no wrinkles in here and somewhere else, but that same acetylcholine is going to also act on other parts of the body, right? So this acetylcholine, you're messing up the release. Now, I just wrote R because this is acetylcholine receptor, right? So this acetylcholine is working with, have many other functions. To not have wrinkles is just a byproduct of one of the functions of it, right? So over time, if you would have that toxin in your body, what would happen? You're doing more harm than good. That's what at least I would think. But some people would still do it. So in some amounts, maybe it is for cosmetic purposes to have a bit of this toxin in your body may be useful. Like in case if you wanted to be like her, not have much wrinkles. But over time, what can be its consequences? Because remember, you need contraction not only in your skin. You need contraction for so many purposes. You need contraction for lifting an object. For everyday purpose, for whatever you do, you need contraction of those muscles. And that acetylcholine is not just, that release of acetylcholine is not being impacted only for these facial muscles, but for several other muscles. And that's probably not what you would want. So that is something that has some real life implications too. So how do actresses use Botox to look younger? And I'm sorry, I'm not biasing it on actresses. I'm sure some actors do it too, but I just have this up here. So how do actresses use Botox to look younger, and what is the mechanism behind it? So when we talk about mechanism in our biology class, we really are talking about what's going on inside the body. What's the physiology behind it, right? So in your own words, you can talk with your neighbor and summarize what we discussed and write it down, and so that I can take a look at it once you go. This should be the 6th question. Okay. So paralysis of facial muscles that prevents the development of wrinkles. Now, when I didn't use the word paralysis in here, you can use the paper. We have a formative assessment. And in case you want to sign the media waiver form, okay, have a pen, pencil. So in here, we talked about paralysis. So one of the downstream effects of Botox is what? Paralysis. You are paralyzing your body. In this case, you're paralyzing your facial muscles. But if the little bit of the toxin is more in your body, it might result in paralysis. And what do you mean by paralysis, really? What is that word? What does that mean to you? What is paralysis? So like you have heard this word before. I'm sure paralysis, you have heard it before. So what is the image that comes to your mind? No movement. So for movement, you need what? The muscles to contract. So preventing muscles to contract would in turn cause what? Paralysis. In this case, it's causing a facial paralysis. So now there are different types of paralysis. This kind of paralysis is also called flaccid paralysis. Again, this is something for general knowledge. You don't need to go too much into the deep. But know what is the the action, the overall action, like how this is important, that is important, and also what is the mechanism that's important. What is really going on in the body? It's important. Now we go to the next part, which is senses. So we kind of talked about motor neurons. So when we talk this type of neuron, what do you think it was? It's causing contraction of the muscles, right? So what do you think this neuron was? Sensory and motor. Motor, right. So this is motor neuron. Now how about sensory neuron? So what are the 5 senses that we got? It's listed over there. So what is it doing? It's getting the information from the environment and it's sending it to the central nervous system, which is either the brain or the spinal cord. So what is going on in here for taste? In the tongue, we have got taste buds. So if we zoomed into the taste bud, we have the sensory neurons in here, and we have the chemoreceptor cells, which is going to send the action potential to the brain. So this also has those channels, ion channels, and that helps with sending the action potential to the brain. Then we have more for taste. This is the ion channel that it talked about. So food is what? It's a chemical, right? So if I have acidic food, if I have sour food, it is going to open a certain channel, because sour means what? It has a lot of H plus ions. So where are my chemistry people or physics people? We have any of them here? No? Okay, so chemistry people should be aware. If not, generic from high school, we know what? That there is more of H plus ions. That means the pH goes what? Up or down? Down, right. So what is the pH 7? What does it look like? There's not too much of hydrogen ions or too less of it. It's just the right amount which you see in the water. But if it goes down, then the pH rises. So in here, if you have sour, something acidic, the H plus ions goes up. So H plus ion is also positively charged, and that means the electrical current being transmitted, you're changing that scenario. So the ion channels change, and that's how the information is going to be transmitted from the taste buds to the brain. Then there is smell. So we have chemoreceptors in the nasal cavity, and then you have the action potential being transmitted to the brain. Oops. So the way it works is we have the cellular response and ion channels. The positively charged ions comes inside. We have the smell receptor proteins. The chemoreceptor proteins corresponds to a particular smell, can be stimulated by airborne molecules. So there are chemicals in the air which is going to stimulate those ion channels to open, lets in ion, then they have an action potential which gets transmitted to the brain. Make sense? So, any question regarding the special senses so far? Now, dogs have as 40 times as many smell receptors than a human, hence their keen sense of smell, which gives them tremendous proficiency at detecting drugs or explosives. So this is, again, something which is different between a dog and a human. Mostly, when you have something more of it, so like in this case, you have more of smell receptors, right? Sometimes it means a compromise with other areas of the brain. So in this situation, what we see is the dogs have a very good sense of smell. Why do you think dogs have evolved to have more sense of smell? Because it obviously served a purpose, a function for them. So they had to detect, a prey or whatnot. So they have a better sense of smell than human, because our mode was more, we were more plant-based rather than, hunting down more animals. But even if it was true that we had a more hunter-gatherer system, we probably had a different ultimate objective. So think of it like if there is a certain function, that's because the structure is there to support it, and it also goes the other way around. The structure is there because it supports a certain function. So which in this case for the dog is you have a heightened sense of smell. Next, the human eye. So in the human eye, what we have is we are able to detect colors. So do you think we get to see everything? That's all the stimulus around us. Do you think we get to see everything? No. We do have blind spots where we can't see. What is a blind spot? It is the place where you have no receptor whatsoever. So at the blind spot, you have no receptor. So what are the rods and the cones? These are the photoreceptor cells that are found in the retina, so the back of the eye. And you see the optic nerve over here, which takes that information from the environment and then sends it to the brain. So the light comes in, the eye lenses readjust to the light, and image is formed on the retina over here, and then it would be transmitted via the optic nerve. So the place where the optic nerve comes together on the retina, that is where you have the what? blind spot. Yes. So the blind spot is the place where we don't see an object. So if there was an image which is formed on the blind spot, we won't be able to detect it because we won't be able to send that information to the brain. So how do the photoreceptor cells work? Pretty much similar. The sodium channels which are located within the photoreceptor cell membranes are open, allowing the photoreceptor cells to continuously release inhibitory neurotransmitter. So this acetylcholine neurotransmitter that we talked about, do you think it is excitatory or do you think it is inhibitory? Excitatory. Why? Because it has the potential to excite those muscle cells to contract. But they can be also inhibitory neurotransmitters. So the sodium channels which are present in those photoreceptors, they have got those ability to pass through inhibitory transmitters. The inhibitory transmitters reduce the ability of the adjacent neurons to excite the neuron that signal the brain. So think of it like there is somebody talking here and another person talking here. So the person in the middle cannot listen to what is going on. So there is an instructor in the front and there are two other people in a row who are talking. So you are trying to receive the information, but you can't because of the noise around you. So it works pretty much the same way. So in here, the inhibitory neurotransmitters reduce the ability of the adjacent neurons to excite the neurons that signal the brain. So you are the neuron which would have otherwise sent the information to the brain. But because of adjacent neurons, you're not able to do that. An action potential is not sent through the optic nerve and light is not perceived by the brain. So that is the way it works in the dark. So in the dark, it is something where you have these noises, the other extraneous noises, which doesn't allow the ones which were in the light to fire. So in the dark, it works the opposite way. We have the inhibitory responses. So in the light energy causes the light-sensitive molecules to change shape, closing sodium channels in the photoreceptor cell membranes. The closing of the sodium channels reduces the amount of inhibitory neurotransmitters, so you're not getting to inhibit anymore. So no longer inhibited, the adjacent neurons excite the neurons that signal the brain, and action potential is now being sent through the optic nerve and light is perceived by the brain. So in the dark, there is a lot of noise around. But in the light, the signal is pure. There isn't much noise around. And now the signal can be transmitted to the brain, which is, when I say signal, I mean the action potential. Hearing, so hearing, as you can see in this organ, it has got, some receptors as well, so which are called in the hair cells. So when those hair cells bend, it's like a physical motion, a mechanical motion, which would change so those vibrations are going to change the amount of fluid which is going through the inner ear, and then the neurotransmitters cause the auditory neurons to fire an action potential, and the signal is passed to the brain where it is interpreted as sound. So there is some noise. So imagine this is the movement of the air around you, and that helps the movement of the bones in the inner ear. And that information, that mechanical stimulus is being now transmitted as electrical stimulus to the ear, the nerves in the ear. And once they get the action potential, then those neurotransmitters are going to, give that information via the postsynaptic neuron and send that information all the way to the brain. Touch, we have mechanoreceptors or thermoreceptors or pain receptors. So it's sensitive to pain. So one thing I would like to mention here, when you have something spicy, even though when we think of spice, we really think of taste, right? But it's not really taste. It's one of these pain receptors. So that is actually a touch receptor. So when that pain comes in, when you think something is hot, it's probably because your pain receptors are getting excited and you have the sensation of hotness. So next time somebody tells you it tastes spicy, tell them it doesn't taste spicy, it is just hot. So we have pain receptors which is going to activate in the tongue and we get the sensation of spiciness level. So this also pretty much works the same way. We have the the ion channels opening up and the ions come in and it gets, the action potential gets transmitted. Now, what is a phantom limb? So, we have many vets coming in who have lost a limb. So, they sense a sensation, they have a sensation in the limb that was lost. So, it's a phantom limb, right? Because you don't have that limb, but you still have sensations of pain in the limb. How is it that possible? So, that is because of this, neurotransmitter. So, think of it as, mirror neurons which are being activated, and because of which you sense a pain in that phantom limb. Now, there is a way to prevent that. So, what you do is you exercise the other limb, and that would feel that, so all the benefits you give to that good limb would now be transmitted to the other limb, because again these neurons have what? They are all interconnected, and so those pain perception which you had in one limb can actually be elevated by giving some kind of exercise or some kind of treatment to the other limb. So that's something new. It's one of the new frontiers of science which discovered. This was discovered, I think, in the last 5 years. So what special sense system is employed to detect how spicy the food is? We kind of just discussed. So you can write that down. I would want to see this, so I have an idea. So what we will do is at the end, after you have written all the responses, at the back, write what were the strengths of the presentation and what were the weaknesses, so I can improve. And that would really help me, and I'll appreciate that. So once you have written down the answers to the question, could you please write at the back what are the strengths and weaknesses, so I can employ that in my teaching strategies, future teaching strategies. Okay, so we have it here. The sensation of pain is what you feel when you eat some spicy food. So now we're going to move a little bit into muscles. Now remember, I talked to you about the nervous system. Okay, so a system means it employs a lot of tissues. So it is not just the nervous tissue that is employing. Now I told you that it could be a motor neuron. Yeah, sure, go ahead. No problem. So in this case, we had a motor neuron, and then it went the synapse. Across the synapse, you had a chemical transmission, and it went all the way to the postsynaptic muscle cell. Now, we need to know what type of muscles, because when that motor neuron is, when we learned about the somatic nervous system, which is voluntary, it has the ability to affect a certain kind of muscles. What do you think they are? They are the skeletal muscles. Even though there are other muscles that are present in our body, the cardiac muscle system, muscles, and the smooth muscles. So usually they are the job of the autonomic nervous system or the involuntary system to coordinate them. So I will just mention briefly, cardiac muscles are in our heart, and then we have smooth muscles, in the inner linings of the body, for example, in the stomach. And then we have got the skeletal muscles, in the arms and the limbs, which helps us moving our arms. So that's a lot of voluntary movements. So the somatic nervous system is usually impacting on this skeletal muscles. So what are the function of the skeletal system? In brief, it just helps with movement. It protects the inner tissues, and it is a place for cell production. So the red blood cells are formed in there. It's a mineral reservoir. So elements such as calcium are released from or stored in bones. So what happens in elderly women is, as they grow older, there is a lot of bone loss. So that's because this calcium which was stored in the bones is now being released even more into the blood, because their blood calcium has fallen down. So that's why we do study the muscles and nervous system together, because one impacts the other. Now, when I say elderly women, so what is there in elderly women which have them to, which causes this condition, loss of calcium from the bones? What do you think is different in elderly women? What is it that was present before and is now not present? So we are talking about postmenopausal women, right? So what happens in postmenopausal women? Have you heard about estrogen before? Yeah. So estrogen is what? It is a neurohormone actually. So when that hormone goes down, this would cause a release of calcium from the bones, and the bone starts becoming brittle. So it's all related. That's how the nervous system is related to the muscular system. So if we don't understand the nervous system, we don't understand hormones. And this hormone is what? It's a type of a neurotransmitter. And therefore, we would, it's all connected. So it's important to know both the skeletal muscle system and the nervous system. So here are just examples of how the skeletal muscles generate movement. So we have the origin point, biceps and triceps, which are the muscles. So everything in yellow is the bone, and everything in red are the muscles. So there is an insertion point and there is an origin for the muscle. So as you can see, the muscle has got two ends to it. Usually the origin and the immovable one would be the origin, and the insertion would be, the other way. The immovable one would be the insertion, and the movable one would be the origin. So we have two sides to which the muscles are bound. So there are different types of skeletal systems. So if you were thinking of this little animal, they have a hydrostatic skeleton. So this is a fluid-filled cavity with sufficient rigidity so that the muscles surrounding it can generate movement. So it's not that they don't have it, but they have a different type of skeleton than what you would see in other, in a human body. So human body has an endoskeleton. So in here, you have another animal which has an exoskeleton. So we have what is present in an exoskeleton is the rigid outer covering that supports and protects an animal's body. The endoskeleton has an internal support structure which is composed of hard mineralized tissue. So what is important here is how do nerves instruct skeletal muscles to contract. So this junction where you see a neuron and a muscle is called a neuromuscular junction or a motor end plate. So whenever there is an impulse being transmitted, I talked to you about, well, when I drew it, this is what I was trying to draw over here. So what you see in here is called a sarcomere, which is the contractile unit. Remember we talked about this when we talked about buttocks? So this is what? It's the contractile system which when contracts produces a movement. So the muscles are what? They are both excitable and they are contractile. So the nervous system was just excitable, and that's often a quiz question. The nervous system is just excitable, whereas the muscular system is both contractile and excitable. So whenever this, at the neuromuscular junction, you have an information, that information passes down to the muscles, which are made up of contractile units called sarcomeres. Within that sarcomere, we have two different types of filaments or proteins. The thin filament is called actin, and then there is the thick filament which is called myosin. So during contraction, what happens is you see this jagged line. This is called a Z line. So from one Z line to another Z line, we have one sarcomere. When the two Z lines come close to each other, that means a contraction has happened. Okay? So what is happening is the Z lines are pulled closer to each other, and this is what? This is the thick filament, right? So when it is being pulled closer to each other, what happens is the thin filaments overlap completely with the thick filaments. So we have the thick filaments sitting right in the middle, right? And we have thin filaments in a relaxed state. Now during contraction, what happens is the two thin filaments come close to each other. So now what we have, we don't have this, zone where it is just thick filament. So just so you know, this zone is called the H zone. The H zone is where we see only the thick filament and not the thin filaments. So during contraction, what happens to the H zone? It goes away, right? So if it is moving closer to each other, this zone goes away. So during contraction, the two Z lines come close to each other, and after the contraction, what it has to happen is immediately relax. What would happen if the muscle remains in the contracted stage? Fatigued, right? So what happens in a dead body? Have you ever seen, I hope you don't have to, but it is like in a dead body, we have something called a rigor mortis. How many of you have heard about it? Oh, many of you. So what is that? It's a repeated contraction. Why do you think that happens? They're dead. So what if they are dead? Yes. Right, so there's no oxygen getting. Why do you need that oxygen? To have that action potential, because action potential, whenever this event is happening, it is very energy expensive. This event requires ATP sometimes. So I talked to you about channels. I didn't go too much into the details, but this type of channel is a ligand gated channel, whereas the channels which are opening in here, they are voltage gated channels. Okay, again in here the channels opening are voltage gated. Usually when there is no electrical current coming in, when there is no stimulus coming in, we have other type of channels that is open. It's called the leaky channels. So if there is no energy required, there is always some leaky channels out there. But in addition to the leaky channels, we have these voltage gated channels that act upon when there is that stimulus coming in, that is the voltage difference being there. Now there is also an ATPase activated channel, which I didn't mention before. So we have another type of, so we have leaky channels, then we have voltage gated channels, we have ligand gated channels, plus we have something called a sodium potassium pump. So this is what happens. So imagine, so these are channels. Now what I'm going to talk about now is a sodium potassium pump. Now which is more inside, sodium or potassium? Inside the cell? So if this is a cell, inside of it, I told you that it is mostly negative, right? And outside is mostly positive. Now inside of the cell, which is more abundant, sodium or potassium? We have more of potassium in the inside and we have more of sodium in the outside. So what is the usual state? The usual state is for this sodium to come inside and this potassium to go outside by the laws of diffusion, which doesn't require any additional energy. So what is the law of diffusion? Movement of anything like ions in this case from a region of its high concentration to a region of its low concentration. So when that happens, what is the expected flow? So this sodium, you expect that to come in, and the potassium, you expect that to go out, right? Now in the case of this ATPase activated pump, where there is energy involved, like she mentioned, there is no oxygen in a dead body, right? So that oxygen is required for what? For energy. That means when you breathe in, you have metabolize food and you generate energy, right? So these ATP is nothing but packets of energy. So in here, the ATPase sodium potassium ATPase pump, the opposite thing is going on. So sodium, 3 sodium ions are going to go out and 2 potassium ions is going to come in, which is opposite to what we just saw. Now you see the difference in the number, this is 3 and this is 2. That also has something to do to make the inside more negative. Why? Because 3 positive charges leaving, 2 positive charges coming in. So net what? Inside is negative. So this is what is the sodium potassium pump. Now in the case of a dead person, we don't have this sodium potassium pump anymore. But in there, this sodium potassium pump is required to what? To make the cell active again so that another action potential can come in, right? So think of this now. We have a contraction going on, and that contraction, after that contraction, we need relaxation, which means we need to go back to this repolarization state to relax it, right? For depolarization, you have contracted it. Now you need the repolarization to relax the muscle, correct? Now in that contraction phase, you need, in order to relax completely, you need those sodium potassium pumps, and you don't have them anymore. So what happens is you are now not having the ability to relax the muscle. And when you cannot relax the muscle, and I'm giving you a very simplistic way I'm explaining it, there are more details to it, but this is at least, the basic knowledge which you might find it useful to explain it to someone, who is asking why there is repeated contraction in the case of rigor mortis. So so this is exactly, this PowerPoint slide tells you exactly what is going on for contraction. There is a detachment between the myosin and the actin. Then they are going to reach towards each other, they are going to reattach, and then they are going to pull back. So a relaxed sarcomere looks like this, and a contracted sarcomere looks like this. So there is an ATP which is involved in this process. So rigor mortis really is happening because of this reason, that during that contraction you require that, not just during the contraction but also during the relaxation process, you require ATP. Now when there is no ATP present, there is no more relaxation going on, and that's why in rigor mortis we have repeated contractions. So what are the different type of muscle fibers in your body? They could be either fast or slow. So what happens in the case of a sprinter? So in the case of a sprinter, what do you need for a sprinter? What do you think a sprinter needs? He needs quick energy resources, right? So that energy would be coming from what source? So there are two types of metabolism. That could be anaerobic fermentation and aerobic. So if you have aerobic respiration, what happens is there is a lot of oxygen which is produced, but that happens really slow. So in the case of that slow fibers, you see a lot more of the aerobic respiration, and so the slow fibers are usually going to be having red cells, because there's more oxygen, and more oxygen means it binds more to hemoglobin, and that means you have a red color. So if you have a slow fiber, that would be red, but if you have a fast fiber where you see anaerobic fermentation, there you have white muscles, more of white muscles in there. What is anaerobic fermentation? It is the situation where you have, where not much no oxygen is being required, but it still produces a little bit of ATP. So you have bursts of energy or ATP produced during that state. So how do you weight lift? Which type of neurons facilitate this movement? So what is happening for weight lifting? So you'll be doing, let's say, a bicep curl. So that is engaging what type of neurons do you think? Is it a sensory or is it a motor neuron that it is? Motor neuron, yeah, so we got it by this time. So how do you weight lift? It is because the muscles are contracting, that's how you can pull the weight. So contraction of the muscles being facilitated by the motor neuron. So using what type of muscles? Obviously skeletal muscles, because those are voluntary in nature. And motor neurons talk to the skeletal muscles at the motor end plate or the neuromuscular junction, and that facilitates this movement. So the last segment of this presentation is the principal regions of the brain. So in the brain, we have the cerebral cortex, which is involved in abstract thought, problem solving, and language. Thalamus, which receives sensory input and relays some signals to the cerebral cortex while blocking others. Hypothalamus regulates many fundamental drives, including hunger and thirst, sexual activity, and maintenance of body temperature, controls the hormone secretions of the tiny pituitary gland. Now, I'm first going to talk about the forebrain. Now, those are the different structures in the forebrain and the functions they do. So think of hypothalamus like the soul of the body, because it really maintains homeostasis or balance. So if something goes wrong, it is the hypothalamus which is going to bring back that balance. So where can you lose balance? So it could be a water loss, it could be a temperature. So you want to maintain a temperature at which you're comfortable, right? We are exotherms, and we would not exotherms, we are endotherms, right? So we would like to maintain a certain body temperature. We want to maintain a constant body temperature. So that is that balance, fine balance, which is attuned by the hypothalamus. Then the cerebral cortex has more higher order functioning, and the thalamus is going to relay the sensory information from the outside world to the cerebral cortex. What about the midbrain? It filters and evaluates the motor and sensory neuron signals. So, it is right over here. So, this part is the midbrain and this was the forebrain. Then we have the hindbrain, which has different parts. The cerebellum coordinates motor activity, the pons has pathways for motor and sensory neuron signals, and the medulla has pathway for motor and sensory neuron signals, which regulates basic physiological functions such as respiration, heart rate, and digestion. So, remember I talked to you about the sympathetic and the parasympathetic centers in the brain. Now, these are the centers which control the rate of heartbeat. So, the heart, the cardiac muscles have the ability to generate its own action potential. So, it is myogenic. But if you have these signals sent from the brain, it can regulate the heart rate. So, when I give you the example of a dog chasing, so this center of the brain, the medulla, is going to send that information through the spinal cord. It is going to send it back to the heart, and then what would happen? The heart rate is going to go up. So, when you are excited, your heart rate goes up, right? That is because of the center in the brain which is controlling the heart rate. So, it has the ability to change the heart rate. And same with respiration. What happens when you are excited? You breathe in fast, right? When you get angry, you breathe in fast. So, that is because of these centers in the brain which has the, the autonomic nervous system responses being controlled by these centers in the brain. Also, the cerebellum coordinates motor activity, which means what? So, you learn swimming when you are young, or you learn piano when you are young. So, when you are older like me, would I be able to learn swimming or piano that easily? Most likely not. The reason is because the fine motor control is being done via that cerebellum, and it forms those connections from in the developmental years. As we grow older, we do not have that much of ability to go back and have a good control and have good synapses in the cerebellum. So, it is difficult for us to learn a new motor skill. So, complex movements, so like after complex muscle movements are learned, such as the precise sequence of knitting, you do not really need to have additional attention to do that, because it is already wired in your cerebellum. So, you can knit and talk on the phone. Or maybe people who are good at swimming, they already know it, their body knows it. How do they know that? It is because that information has been restored in the cerebellum. And now whenever you need it, it is already there. So, it is not difficult for them. For me, if I have to learn knitting now, I have to pay more attention to that task and learn it before I get used to it. Now, this guy, Phineas Gage, his frontal cortex got altered because of an accident, and I wouldn't say thanks to that incident, but I would say that was one of the ways we learned about what is the function of the frontal cortex. So, regions of the cerebral cortex, we have the frontal lobe, the temporal lobe, parietal, and occipital. Easy to remember: frontal in the front, temples, we know the temples, right? So, temples over here, and then we have the parietals in here, and then the occipital at the back. So, speech centers in the brain called the Broca's area. So, Broca was a doctor, and he had a patient, multiple patients who came with aphasia, means couldn't speak. So, after they died, obviously, we were able to look into their bodies and into the brain specifically, and we found out the Broca's area, which is the area which coordinates the muscles which is necessary for speech production. There is another area for speech called the Wernicke's area, which is responsible for language comprehension. So, there is a Broca's aphasia, inability to produce speech, so it is slurry. And if there is something wrong in the Wernicke's area, it is for language comprehension. So, if you speak a foreign language to me and have something wrong in the Wernicke's area, it is even more difficult for me to understand what is going on. So, Wernicke's area is more for the language comprehension, whereas for the speech production, it is the Broca's area. Memory centers, there is an amygdala and a hippocampus. Hippocampus is mostly storing the short-term memories, and it is important for retrieval of memories. Emotional memories like fear or anger, hatred, love, not love, it is fear mostly, not love, no positive emotions, but mostly the negative valence emotions would be stored in the amygdala. So, the fear response is already, so if you give a shock to a rat all the time, then whenever, even if it sees something closely related to what you gave it, that if it associates, feeding with shock, then it would not feed itself anymore, because it's like it's thinking, oh, it's afraid of that response, right? So if you're gonna associate one activity, which is most likely not feeding because it has a positive valence to it. Let's say if you associate red color with shock, then the rat is going to remember that, and its amygdala is going to be responding because it has a fear response, and whenever it sees that red color, it would not want to come close to you, or it wouldn't. It would be afraid. So reuptake inhibitors, I think this is something very important. We're kind of almost there, but this is an important topic, guys. What is a reuptake inhibitor? Some of us do take Prozac for depression. So what is happening is, we talked about the neurotransmitters in here. The neurotransmitter that I talked about mostly was acetylcholine. But there are other neurotransmitters like dopamine, serotonin, and those are, specifically dopamine is an inhibitory neurotransmitter. So a reuptake inhibitor is, there are ways in which the neurotransmitter can go back, okay? But if it is a reuptake inhibitor, that means it stays longer in the synapse. So serotonin and dopamine and norepinephrine is known to affect mood. So if you are going to have a reuptake inhibitor, what happens is, in the synapse, you are having more of those neurotransmitters stay. And having them more means you have the ability to control your mood, because earlier it was just thought that if you don't have enough of those neurotransmitters, because of which you have those mood swings. So you can take a reuptake inhibitor, and now you can enforce that neurotransmitter to stay in the synapse, and that has the ability to again alter your mood. So I'm not going to go over the slide, but pretty much I explained what the mechanism is. It's the way that the presynaptic neuron is not able to take the neurotransmitter. Again, remember, if you take a dopamine molecule directly, that doesn't have the ability to cross the blood-brain barrier. So you need a precursor. You need to take a precursor, and then that way it would go. But if you have a reuptake inhibitor, then naturally whatever the body produces, that dopamine, you're just enforcing it to stay longer in the synapse, and thereby affect downstream processes, which has the ability to alter mood. So depression, ADHD, these are all connected to what we learned, because these are all impacted by the neurotransmitters, and these neurotransmitters are going to also alter the way we learn, and they produce a physiological response that might be responsible for a personality type. The other neurotransmitter or stimulant is caffeine, and the way caffeine works is it has the ability to stay longer and just fire those neurons more, and it also acts in the same area which controls mood, the area in the the prefrontal cortex. And also there is alcohol, which is another stimulant, which also messes up with the neurotransmitters in the brain, and also it has the ability to affect the cerebellum. So that is why cerebellum, which is also involved with balance, is lost because of the intake of alcohol. So why do you drink coffee in an 8 a.m. class? Which part of the brain is being affected? Coffee is the stimulant, which affects the reward center in the prefrontal cortex. So drink more coffee, you want to drink more coffee because it's activating the reward center. Guys, thank you so much for listening to my presentation. I would really appreciate if you would write down at the back of this paper what do you think about what I could improve and what you liked about the presentation. And these answers would be already there in the PowerPoint. So if you go to Blackboard, you should be able to access them. You can, yes, because at the back of it, I need to see both of them. Thank you so much.